Hello my dear students, welcome back to Biology at Priyajit. In part 3 of Kingdom Monera, we are going even deeper into the incredible diversity of prokaryotic life. In this class, we will explore some very special and unique groups such as Cyanophyceae or blue-green algae, Mycoplasma, Actinomycetes, Spirochaetes and Rickettsia. These topics are extremely important for class 11 biology concepts as well as NEET preparation, and they will help you build a sharper and more conceptual understanding of Monera. So get ready to discover some of the most fascinating and unusual microorganisms. Let's continue our journey into the microscopic world. Blue-green algae, BGA or cyanobacteria, or you can call as Cyanophyceae. They're called as blue-green algae or Mycoplasma and Cyanophyceae, but in 1978 International Code of Nomenclature of Bacteria ICNB proposed them as cyanobacteria. Cyanobacteria are unicellular gram-negative prokaryotes basically found in aquatic environments or hot spring, marine water, below the iceberg, freshwater, lake, wetland, etc. They are either symbiotic with other organisms like marine planktons, fungi, liverworts, fern, angiosperms, small protozoans, sponges, corals, mammals, etc. or photoautotrophs having pigments like chlorophyll, phycocyanin, phycoerythrin, etc. like the plants. Cyanobacteria basically shows chromatic aberration as their body color changes in response to various wavelengths of light. Red Sea is red colored due to the presence of a red colored cyanobacteria or Trichodesmium erythraeum in polluted water bodies. Cyanobacteria forms blooms. BGA or blue-green algae are surrounded by a mucilaginous sheath. A few cyanobacteria like nostoc species, Aramina species, fix nitrogen with the help of a thick-walled special structure called as heterocyst. This sheath is made up of mucopolysaccharide made up of pectic acid. The cell wall of BGA is also bilayered. Outer wall is made up of lipopolysaccharides and their inner wall is made up of peptidoglycan. The cell membrane of blue-green algae is also made up of lipoproteins. Cytoplasm of BGA contain following structures like thylakoid or chromatophore, which are the membrane-bound structure containing photosynthetic pigments. Inclusion bodies: BGA store its food in the form of a alpha granules. Alpha granules are made up of cyanophycean starch. It is structurally similar to glycogen. Gas vacuole, which is membrane-bound structure and found in blue-green algae. Purple and green photosynthetic eubacteria. Nucleoid or double-stranded circular naked DNA, 70S type ribosome, etc. In BGA, reproduction is done by two main processes: vegetative reproduction, asexual reproduction. Vegetative reproduction is performed by binary fission. It is the most common method of reproduction in prokaryotes such as spirulina. Fragmentation performed by hormogonia or hormocyst formation. Filamentous prokaryotes reproduce by this process such as oscillatoria. Asexual reproduction, it is a method of protection to overcome unfavorable conditions, that is actinic formation in nostoc. Economic importance of blue-green algae. Useful activities include it gives fertility to sterile alkaline soil and acer soil. Blue-green algae secretes acidic chemical which decrease the alkalinity of soil. Some BGA are used as green manure. Some BGA secrete toxin which inhibit the growth of mosquito larvae in water. Harmful activities: water bloom, BGA that grow in water tanks pollute the water, such as example oscillatoria. Mycoplasma. Nocard and Raux in 1898 discovered the mycoplasma. They are also known as PPLO or pseudopneumonia-like organisms, or MLO, mycoplasma-like organisms. They are polymorphic, gram-negative, simple, the smallest prokaryotes of 0.5 to 0.15 micron without cell wall. Their cholesterol enriched cell membrane is composed of polysaccharides containing acetyl glucosamine. Saprophytic or parasitic, ETS, electron transport system, is absent in their body. They are obligatory anaerobes. Binary fission is their principal mode of reproduction. They are pathogenic to plants, human and other animals. Mycoplasma pneumoniae causes pneumonia in human and other animals. Mycoplasma salivarium, Mycoplasma oral causes upper respiratory tract infection in human and other animals. Mycoplasma bovigenitalium causes infection in the genital organs of the cattle. As they have no cell wall, they are insensitive to penicillin, and Mycoplasma gallisepticum is the smallest living organism. Actinomycetes are earlier they are called as ray fungi. They are gram-positive, saprophytic, aerobic, mesophilic prokaryotes. They have similarities with the mycelium of fungi, so they are called as fungi-like bacteria. They are different from other bacteria in their ability to form a mycelium. They can produce antibiotic and pathogenic to human. Examples include Actinomycetes, Streptomyces gracias, Streptomyces aureficiens, etc. Spirochaetes, they are gram-negative, coiled, elongated, flagellated, heterotrophic bacteria found in wet, muddy places. They are pathogenic to human. Treponema pallidum causes the disease like syphilis, Beazell and yaws. Leptospira causes relapsing fever, and Borrelia species causes infective jaundice. Rickettsia are gram-negative, rod-shaped, pleomorphic parasitic prokaryotes. They have many similarities with virus. They are also called as the intermediate form between virus and bacteria. The flagella, pili, mucilage capsule are absent. Binary fission is their principal mode of reproduction. They live in the intestine of arthropods and causes infection in human. Rickettsia typhi and Rickettsia museri causes murine typhus disease. Rickettsia rickettsii causes Rocky Mountain fever. So we can conclude that in part 3 of this chapter, Kingdom Monera, we have explored some of the most unique and specialized groups of prokaryotes, including Cyanophyceae, blue-green algae, then Mycoplasma, Actinomycetes, Spirochaetes, and Rickettsia. We understood their distinct characteristics, structural features, and ecological contribution and medicinal significance, which further deepens your understanding of microbial diversity within Monera. If you found this class helpful, don't forget to like, share, subscribe, and share with your friends. Now get ready for the next part. That's the next Kingdom Protista, where we will continue exploring even more exciting concepts in biology. Thank you.